So where we're headed today is we're going to talk a little bit more about the generation of excitatory postsynaptic potentials. We'd gotten to the point last time where we had discussed the ligand-gated channel, that is, those channels that are operated by a neurotransmitter. And we talked about them in terms of those in the peripheral nervous system being predominantly opened by the neurotransmitter acetylcholine, and that in the peripheral nervous system, the acetylcholine was largely disposed of by the enzyme acetylcholinesterase. But in the central nervous system, the predominant neurotransmitter is glutamate, the simple amino acid glutamate. And glutamate binding to glutamate receptors opens the channel by, in a similar way, swinging open a gate so that sodium can come in. In both cases, whether we're dealing with acetylcholine in the periphery or whether we're dealing with glutamate in the central nervous system, it's almost entirely opening ligand-gated ionotropic channels that are permeable to sodium. And so we'll talk a little bit about that. And remember, if you have an increased permeability to sodium, that's going to be a depolarizing potential. So you're going to depolarize the membrane. So these, by depolarizing the membrane, this is going to be excitatory. Just in the way we used those stretch-activated channels to excite the membrane by pulling them open, by deforming the membrane, we're now going to do something very similar here at the synapses using a ligand-gated ionotropic channel. By opening that ligand-gated ionotropic channel, we'll let in sodium. This is going to produce a small excitatory postsynaptic potential. These small potentials, which in the peripheral nervous system tend to be, in the neighborhood of a half a millivolt or less, will then summate at a site called the trigger zone. Again, remember back to the idea of opening those stretch-activated channels. The similar story here is that we're now going to open ligand-gated channels, and the number of ligand-gated channels opened will be proportional to the amount of neurotransmitter released. Since we're pretty much limited in the amount of neurotransmitter we can release in each case because of the number of neurotransmitter molecules in a vesicle, this means to get summation, we must open many, many vesicles in order to have those small potentials generated on the postsynaptic membrane. And then as they propagate over the membrane of the input region, they're now going to summate at the trigger zone. And again, we're going to see that the trigger zone is characterized by a site where we cluster together voltage-gated sodium channels. That will then initiate an action potential, will propagate down the conductal region to the output region of that cell. But as we think about this, what I really want to emphasize today is the idea of how these potentials are summating and how the characteristics of the membrane in the input region will affect the summation. So we're going to talk a little bit about the idea of leak channels. Again, back to the very idea we initiated a few days ago, where we talked about the role of leak channels allowing positive charge to escape from the cell. This same story is going to pertain here, but now we're going to confine these leak channels for this part of the discussion to the regions of the input region of the neuron. And what we mean by that is the regions where we have an enrichment of those ligand-gated ionotropic channels responding to the release of neurotransmitter. That will generate an excitatory postsynaptic potential. But the excitatory postsynaptic potential is, in fact, the summation of many, many synaptic sites. And those many, many synaptic sites will be summating, but we have to think about the potential for these, the possibility of these potentials to decay by the time they reach the trigger zone. There's the ability of these potentials to reach the trigger zone relatively unchanged is largely dependent upon this term R. We're going to see a voltage is going to be produced. The voltage is going to result from the current times the resistance. Simple idea. Well, this current is going to be fixed because for an individual synapse, the amount of current won't change very much. It's going to be produced, it's going to be limited by the number of channels that open, the number of neurotransmitter molecules that are released. And so that's going to be pretty fixed. So we can have more or less a fixed voltage. But now what's going to vary, for our purposes here, is the membrane resistance, the number of holes in the membrane. And the greater the number of holes in the membrane, the leakier the membrane, the more likely it is that that charge is going to leak out across the membrane before arriving at the trigger zone. So we'll just try to think our way through the idea of variations in the membrane resistance. But then the next step is to think about how this happens in biology. Because one of the major ideas in biology is going to be inhibition. If you take some more neurobiology courses, you're going to find that the role of inhibition in the central nervous system, in all central nervous systems, seems to be predominant. We like to tell the story from the point of view of excitation, bringing things to threshold, firing action potentials. But without inhibition, the entire system runs out of control. So we'll spend a little bit of time here talking about inhibition. We're going to emphasize here the idea of GABA and glycine-operated channels. These two neurotransmitters, GABA and glycine, work similarly to increase chloride permeability. Now, everything up to now has been based on increasing a sodium permeability. And now we're going to increase a chloride permeability. And under most circumstances, the chloride equilibrium potential is more negative than the resting membrane potential. So this produces a hyperpolarization. We put more negative charge into the inside of the cell. If the inside of the cell is more negative, we'll get a hyperpolarization, an inhibitory postsynaptic potential. But there's a little bit more sophisticated way to think about this beyond just the potential. And that is to think in terms of the permeability and in terms of the equilibrium potential. And the idea that the membrane potential seeks the equilibrium potential for the ion whose permeability is dominant. So if the dominant permeability is going to be to chloride, it doesn't matter where we set the equilibrium potential. As long as we open these chloride channels, it will clamp the membrane potential at that equilibrium potential for chloride. And we'll talk in some detail about that.